Special connective tissue, cartilage, general characteristics. Cartilage is a special type of connective tissue which has a rubbery rigid matrix, not traversed by blood vessels, nerves, or lymphatics, and entirely nourished by diffusion. Structure. Cartilage contains characteristic cells which are either immature chondroplasts or mature chondrocytes, arranged either singly or in groups inside their lacunae called cell nests. Cartilage contains also matrix and fibers. The matrix is a complex mixture of glycosaminoglycans responsible for the basophilia of the matrix, formed of hyaluronic acid and chondroitin sulfate. In the matrix, there is also structural glycoprotein, mainly chondronectin. The fibers present in the matrix are collagen fibers type 2 and elastic fibers. Types or classification of cartilage. Cartilage is classified into 3 groups according to differences in the abundance and type of fibers in the ground substance or the matrix. These groups are hyaline cartilage, yellow elastic cartilage, and white fibrocartilage or the fibrous type. Sites. The sites for presence of cartilage differ according to the type. The sites differ in hyaline cartilage from that of yellow elastic or white fibrocartilage. Cartilage growth. Cartilage grows by 2 methods: interstitially in length, only in young cartilage, and appositionally in width. Interstitial growth occurs in young cartilage from division of young chondrocytes, thus leading to increase in the cartilage length, while appositional growth occurs from division of the chondrogenic cells in the perichondrium, differentiating into chondroplasts, thus forming a cartilage layer around the periphery of the existing cartilage, thus increasing its width. Functions. Cartilage, together with the bone, forms the skeleton of the body. Cartilage helps easy movements by covering the articular surfaces of the bone by articular cartilages at the joints. Cartilage keeps the respiratory passages patent by forming a cartilaginous ring around the bronchial tree. Hyaline cartilage. Hyaline cartilage is the most common type of cartilage. Structure. Perichondrium is a vascular connective tissue membrane present on the surface of the cartilage. It is made of collagenous fibers and fibrocytes arranged in 2 layers, outer fibrous and inner chondrogenic layer. The perichondrium contains plenty of blood vessels to supply the cartilage with nutrition by diffusion, as the cartilage is non-vascular. Its deeper chondrogenic layer contains chondroplasts, that is, immature cartilage cells, to form new cartilage. Functions of the perichondrium. Nutritive function through its content of blood vessels. Chondrogenic function, that is, formation of new cartilage during growth and regeneration. Also, it gives attachment to muscles and tendons. Cells. Chondroplasts or immature cartilage cells are present in the inner chondrogenic layer of the perichondrium. They show by electron microscope all features associated with protein synthesis, that is, numerous rough endoplasmic reticulum responsible for the basophilia of the cytoplasm, also Golgi apparatus, mitochondria, and secretory granules. They elaborate the specialized extracellular matrix of the cartilage. Once they mature and become imprisoned inside the lacunae, they are changed into chondrocytes. Chondrocytes are basophilic cells present in lacunae. The lacunae are surrounded by a condensed matrix called capsule or tertial matrix. The superficial chondrocytes are small, flat, and arranged parallel to the surface. They are present single in their lacunae. Young chondrocytes can divide inside the lacunae once or twice, forming cell nests. Mature chondrocytes, on the other hand, do not divide. The deeper chondrocytes are mature or old, are large, rounded, oval, or triangular, and arranged perpendicular to the surface. They are found in their lacunae either single or in groups called cell nests containing 2, 4, 6, or 8 cells. Matrix. The matrix or the ground substance of hyaline cartilage appears homogeneous and basophilic, although it contains a mixture of collagen and elastic fibers. But the fibers cannot be seen because they are very thin to be seen by the light microscope. Also, they have the same refractive index of the matrix. The fibers, however, can be demonstrated after digestion of the ground substance with either trypsin or alkalis. Sites. Hyaline cartilage is found in costal cartilages between the ribs, long bones of fetal skeleton, articular cartilages or articular discs at the joints, cartilaginous rings around the trachea and the bronchial tree. Yellow elastic cartilage. Structure. Yellow elastic cartilage is similar to hyaline cartilage, having perichondrium cells, but the matrix contains, besides the usual composition, abundant yellow elastic fibers. Sites. Yellow elastic cartilage is found in ear pinna, external auditory meatus, Eustachian tube, and epiglottis. Fibrous cartilage or white fibrocartilage. Structure. White fibrocartilage differs from other types of cartilage. It has no perichondrium. The matrix contains abundant parallel collagen fibers separated by rows of chondrocytes. Sites. White fibrocartilage is found in symphysis pubis, intervertebral discs, interarticular cartilages, and lips of the glenoid and acetabular cavities.